Today's lecture is going to cover two different sections, 24.2 on gene linkage and 24.3, which is going to cover what happens when we change chromosome number. So gene linkage, each chromosome contains many alleles in a definite fixed order, and they have what we call linkage groups because all the alleles on one chromosome that get inherited together make up a linkage group. So the closer they are together, the better the chances they're going to be inherited together. Sex linkage refers to sex chromosomes. Two traits crosses assume that alleles are on non-homologous chromosomes. So when we looked at the dihybrid cross that we did in the last chapter, we assumed that the traits we were talking about were located on separate chromosomes and they assorted independently of each other in meiosis. Groups of alleles that are linked don't show independent assortment. So that's what we're looking at with gene linkage. In this individual, the alleles A and B are on one member of a homologous pair, and you see this on both of these pictures. Alleles A and B are on the other member, so we have capital A, capital B, lowercase a, lowercase b, and we have it in both situations. The individual is a dihybrid, and when linkage is complete, the dihybrid produces only two different gametes. So we don't have crossing over, and we have the two possibilities being big A, big B, little a, little b. When linkage is incomplete, the dihybrid produces four types of gametes instead due to crossing over. The recombinant gametes occur in reproduced proportion because crossing over occurs infrequently. But you notice at this point where they had crossing over occur that 97% of the time that crossing over did not occur, and this recombinant gametes only happen 3% of the time. So the incomplete linkage is not near as prevalent as complete linkage. During meiosis, crossing over sometimes occurs between non-sister chromatids in a tetrad. The chromatids exchange genetic material. If crossing over does occur, the dihybrid produces four types of gametes instead of two types of gametes. The occurrence of crossing over can help tell the sequence of genes on a chromosome. Crossing over occurs more often between distant genes than between closer genes. So if we look at this pair of homologous chromosomes in this case, and crossing over did occur, the chances of it crossing over happening between Z and G are higher than the chances of it happening between S and R because they're closer together. Let's see what happens when there's a change in chromosome number. First thing we're going to talk about is non-disjunction. Non-disjunction can occur during meiosis 1 when both members of a homologous pair go into the same daughter cell. It can happen during meiosis 2 when the sister chromatids don't separate and both the daughter chromosomes go into the same gamete. This results in what we call monosomy or trisomy when there's a fertilized egg. So we have 3 copies of a chromosome in the fertilized egg instead of 2, or we have 1 copy instead of 2. Here's an example of trisomy of the 21st chromosome. And we have 3 copies instead of 2 copies. So that was due to non-disjunction, and this is a karyotype of someone with Down syndrome. We look at Turner syndrome on the other hand, and this is not here, so there's not a second sex chromosome. So it's monosomy because there's only 1 copy of the X. There's not another X or a Y instead. And this is a female, and both of these mutations are viable. Individuals can live with Down syndrome, and they can live with Turner syndrome as well. In this first example, non-disjunction occurs here in meiosis 2 if the sister chromatids separate, but the resulting chromosomes go into the same daughter cell. So we have them both in the same daughter cell here, and then the egg has 2 copies of that particular chromosome, and when it's fertilized, it has 3 copies of that particular chromosome, where the other side will have no chromosome at all. So if that egg is fertilized, there's only 1 copy of that chromosome, and that would come just from dad. In the other picture, we have non-disjunction happening during meiosis 1, and you can see how this can easily lead to the same situation, only a little bit grander scale because it's more frequent than if it happened in meiosis 2. When it happens in meiosis 1, we end up with 4 cells that, if they're fertilized, have the incorrect number of chromosomes for that particular organism. Normal development of an individual depends on us having exactly 2 of each kind of chromosome. In humans, trisomies are tolerated better than monosomies are. Trisomy 21, or Down syndrome, has a reasonable chance of survival after birth. Chromosome 21 is one of the smallest chromosomes. The chance of survival are greater when the trisomy or monosomy involves the sex chromosomes and not the autosomal chromosomes. Abnormal number of sex chromosomes. A normal XX female, each one of us that have an XX have one X that's inactive, and this is what is also referred to as a bar body. The cells of females function with one X just like males function with only one X. A zygote with one X chromosome without having that extra one and has Turner syndrome can survive. And all the extra X chromosomes then become deactivated. Poly-X females, so this is a female with more than two X's, and XXY males are seen fairly frequently in our population. An extra Y chromosome can also be tolerated. XYY is known as Jacob's syndrome and is due to non-disjunction during meiosis II during spermatogenesis, which is the making of the sperm cells. Let's look a little bit closer at Down syndrome, which is trisomy 21. Most common autosomal trisomy that's seen among humans is Down syndrome. It has easily recognizable physical features, mild to severe mental impairment that comes along with this disorder. The chances of a woman having a Down syndrome child increase with age. So at about age 40 and on up, the chances of having a child with Down syndrome increases rapidly. There's a process called karyotyping that can be used with amniocentesis, or a process called chorionic villus sampling to diagnose a fetus. And it's thought that the symptoms may be due to expression of a gene called the GART gene that causes the disorders of someone with Down syndrome. This is the region of the GART gene on the 21st chromosome. A karyotype was mentioned in the previous slides, and I showed you two different karyotypes, one of Down syndrome and one of Turner syndrome. So it's basically where they would just take a cell, they would kill it during the process of cell division so that the chromatin is condensed into chromosomes. They would take a picture of it and then cut apart the different chromosomes and match them by size. And when they do that, they notice that there are 3 of the 21st chromosome in this case. Let's look at changes in sex chromosome number and what happens. So the presence of Y chromosome, not the number of X chromosome, determines the maleness. There's an SRY gene that produces the testis determining factor on the Y chromosome. So we look at Down syndrome. It affects males or females. The chromosomes that are affected, we have 3 copies of chromosome 21. The total chromosome number for an individual with Down syndrome is 47. 1 out of 40 will experience a spontaneous abortion when pregnant with an individual with Down syndrome. And about 1 in 733 live births will have Down syndrome. A poly-X individual is a female, so we have one that is XXX or has 4 X's. So you can have 47 to 48 chromosomes. These rarely spontaneously abort, and about 1 in every 1,500 have this disorder. Klinefelter syndrome is a male, and they're XXY or XXXY. So you notice we have those multiple copies of the X, but remember those extra ones get deactivated, so they can have 47 or 48 chromosomes. Spontaneous abortions for someone that is carrying a child with Klinefelter syndrome is 1 in 300. Live births, we see about 1 in 800 with this disorder. Jacob's syndrome is a male disorder, and that individual will have a karyotype showing XYY, which is 47 chromosomes. Not sure how many of those that have fetuses with Jacob's syndrome are spontaneously aborted, but there are about 1 in 1,000 born with this disorder. And then Turner syndrome, which is a female, and they are born with just 1 X. So they have a chromosome number of 45 instead of 46. 1 in 18 are spontaneously aborted, and we see 1 in 2,500 live births of those people that have Turner syndrome. Just to give you an idea of some people, famous people with some of the genetic syndromes that we've talked about. The gentleman on the right is Chris Burke, and he was in Life Goes On and a couple of other TV shows that were on back in the 80s and 90s. And this is a picture of him and his wife and his child, and he's a successful actor, makes lots of money. The lady on the left is Linda Hunt, and she plays Hetty on NCIS Los Angeles. She has Turner syndrome. So are there some facial features, some body features that seem abnormal compared to what we consider normal? Yes. Short in stature. Facial abnormalities are just a little bit different. For the Turner syndrome, for Down syndrome, the Mongolian type eyes, the thickened short tongue, lots of facial features that we see and we recognize that as Down syndrome, but does that mean you can be any less of a success or any less of a person? That's absolutely not, because they're different doesn't mean that it's something that should make them so different from everyone else that we see them as an outcast. So just a couple of examples of success stories with Turner syndrome and Down syndrome.